Right now we have Dr. Hide Okeda, who received his MD degree from Nagoya University School of Medicine in 1991. He subsequently completed his internship and residency of neurological surgery at Nagoya University Graduate School of Medicine in 1996. After postdoctoral training at the University of Pittsburgh School of Medicine, he was appointed as tenure-track assistant professor of neurological surgery at the UPSM in 2001. Dr. Okeda currently serves as an associate professor of neurological surgery with tenure, as well as a co-leader of the brain tumor program in the University of Pittsburgh Cancer Center. In 2010, Dr. Okeda was elected to be a member of the American Society for Clinical Investigation, an honor society of physician scientists, those who translate findings in the laboratory to the advancement of clinical practice. Dr. Okeda is on the advisory boards of the Musella Foundation for Brain Tumor Research and Voices Against Brain Tumor Cancer Foundation. He is well known in our brain tumor world for his work with vaccines and clinical trials. Please welcome Dr. Okeda.
Thank you, Laura, for your very, very kind introduction. Thank you, Al, Stan, and Pam for giving me a ride this morning at the airport and also organizing this exciting event. And thank you all for staying for my talk despite this weather condition. And can I get my first slide? Well, this is always my first slide, and this is not particularly the one I made for this occasion. I always use this and introducing my colleagues. And also, I wanted to, I always in my talk and seminars, I emphasize my appreciation to patients and patients' families. I think I am very fortunate. I love research, and I am fortunate working with my wonderful colleagues you see here. I wouldn't have been here without any one of these. And as Laura kindly introduced, my vaccine work actually stemmed from our own laboratory work. So we started from scratch in the laboratory and got translated into clinic. So in that process, there are lots of people who contributed. My wonderful adult neurosurgery, neuro-oncology colleagues, pediatric neurosurgery, neuro-oncology colleagues, my laboratory, past and current laboratory members, and Cancer Institute wonderful immunologists, and industry colleagues, and funding sources, including particularly Musella Foundation for continued research, continued support for research, but perhaps most importantly, I always appreciate the participants and their wonderful families. And another big reason I think myself fortunate is because through my work, I have a bunch of opportunities to see wonderful patients and wonderful families, with truly respectable families who support their family patients for participation into our clinical trials. So having said that, I have been looking forward to talking to you today, and this is truly my pleasure to talk to you today. So conflicts of interest. So one of the peptides that we've been using in our vaccine has been licensed to a company, Stemline, so I have to declare my conflicts of interest. However, the data I'm presenting today, interpretation of data have not been made by myself, but have been made by our entire research team. And scope of my presentation, I'd like to talk about 3 different topics today. First, I'd like to start off with kind of like a background information about cancer vaccine, what cancer immunotherapy is. And secondly, I'd like to talk about my own vaccine trials going on in Pittsburgh. There are a bunch of different vaccine platforms being tested currently for brain cancers, and I hope that every one of those will succeed, and I have tremendous respect to other different approaches. But for the limited time, I'd like to focus on my own vaccine approaches that are being conducted in Pittsburgh. And lastly, I have a new development, which I consider as the potentially dream cancer immunotherapy. So I'd like to touch upon that at the last portion of my talk. So first, cancer vaccine. What's cancer immunotherapy? So as you can imagine, the cancer immunotherapy, the main premise is to stimulate the patient's immune system to attack the tumor cells. And there are 2 fundamental concepts. The first one is called active immunotherapy approach, active immunotherapy, in that we count on the patient's own immune system. So the typical example is the cancer vaccine. So in the case of cancer vaccine, you give vaccines, but the vaccine by itself, in the laboratory setting, if you mix the vaccine material and the tumor cells in the test tube, it doesn't kill tumor cells. It has to go through the patient's own immune system and activation of immune system in patients. On the other hand, there's another concept, different concept called passive immunotherapy. So in that, effector cells, or let's say drug, are made in the laboratory or factory before giving to patients. For example, monoclonal antibody. Even Avastin can be considered this, a part of this category, because antibody is made in the factory or laboratory and given to patients. Or nowadays, there's a lot of attention for the novel therapeutic approach called adoptive immunotherapy, using patients' own T cells, T lymphocytes, genetically engineered to target patients' cancer cells. So in that kind of approaches, if you mix those products with the tumor cells in the test tube, those effector cells can kill tumor cells immediately. So that's a fundamental difference. In my talk for the rest of the time, I'm going to mainly focus on active immunotherapy approach, because many people want to hear about vaccine approaches. So, however, there are some challenges. The immune system is fascinating. The immune system is absolutely fascinating because it has an ability to discriminate self versus non-self. All right? So, so immune system can recognize anything bad coming from outside of the body, viruses or bacteria, while immune system never attacks your own body components. So when it comes to cancers, cancer cells are originally from patient's own body. So immune system is already tolerated, more or less, to the cancer cells. When I started 10, 15 years ago, the hypothesis was that, okay, because cancer cells still contain lots of abnormal substances, abnormal proteins and so forth that are usually not usually made, that are not usually made by normal cells, we may be able to train cancer patients' immune system in a way that immune system recognize cancer-specific molecules so that immune system can attack tumor cells. That was an hypothesis. And fortunately, we have been able to demonstrate there are some antigens like that. So those are the basis, foundations for developing vaccine strategies for brain cancers. And I was going to show our next slide. And in immunotherapy, though, in general, I also recognize that there has been some skepticism against cancer immunotherapy in general. Sometimes cancer immunotherapy has shown promising results in early phase clinical trials, but those results often do not really stand in the subsequent larger scale trials, maybe due to the selection of patients. Cancer immunotherapists may be, selecting favorable patients who are going to do well to begin with anyway. And however, in the last 1 or 2 years, I have been so pleased, started seeing really hard evidence, what's called hard evidence, showing that cancer immunotherapy can work. In other words, in large scale trials, such as randomized phase 3 trials, cancer immunotherapy approaches have started showing the efficacy in such an objective settings. For example, Provenge, this is a vaccination against prostate cancers, has been shown to be effective to some extent and has been approved by FDA. And another approach is in the monoclonal antibody therapy targeting a molecule called CTLA4. So this was tested in a large scale in patients with advanced melanoma. And in order to see 10 to 15% response, however, they also had to see 40 to 50% of serious side effects, mainly autoimmunity. Well, in advanced melanoma patients, well, autoimmunity is certainly not appreciated, but again, this is a matter of risk-benefit ratio. my point is that there is some advantage, there is some objective efficacy, but there's a lot of room to improve. And also, lastly, I want to point out phase 3 study investigating GP100 peptide vaccine. This is melanoma antigen peptide vaccine. When it was combined with standard of care interleukin-2 therapy, that combination has shown significant improvement of survival in melanoma patients compared with interleukin-2 monotherapy. So this is another piece of evidence in a large scale randomized trial showing that, objectively showing that immunotherapy can work. So let me switch my gear a little bit. So let's talk about brain tumor vaccines in our experience. So when I started my brain tumor vaccine work 15 years ago, not many people believed in it. Many people actually ask, hey, the brain has been known to be immunologically privileged. What it means is there are no lymphocytes, there are not many lymphocytes, there are no lymphatic organs in the brain, and there is the blood-brain barrier that limits the migration of T lymphocytes and so forth. However, this privileged status has been quite revised in the last 10 or 15 years. Number one, there is an autoimmune conditions in the CNS. CNS means central nervous system. Multiple sclerosis is one of the very well characterized pathological conditions. But also I want to mention this very interesting human pathology, paraneoplastic cerebellar degeneration syndrome, PCD. So patients with undiagnosed breast cancers or ovarian cancers come to neurology office complaining ataxia, loss of coordination. Those are typical cerebellar signs. So what it is that those patients, again, have undiagnosed cancers outside of the brain that express a protein named CDR2 here. And this CDR2 protein is also expressed in the normal brain cells. So when the same protein is expressed, made in the form of cancer outside of the brain, for the first time, immune system recognizes this protein, and the immune system is activated against this protein and attack the cells that express the same antigen in the brain. So this is a kind of fascinating pathology, and also in a research area looking at the prognostic factors in ependymoma, those people I'm referring to, investigators in University of Colorado, they were not immunologists. They wanted to learn any genetic factors that can predict good versus bad prognosis of ependymoma patients, and found that immune cell infiltrate and immune signatures are the most important significant factors that predict better survival, better prognosis of patients. So immune system really matters for the brain. But a naturally occurring immune system itself may not be sufficient to reject tumors in the brain. So the question is how we can harness the immune responses against the brain tumors. So many people talk about dendritic cell vaccines, and I am one of those who have conducted dendritic cell-based vaccines. So let me go through dendritic cell vaccines to understand the concept of cancer vaccines. So in this schema, from peripheral blood, we take peripheral blood samples, peripheral blood mononuclear cells, that is now abbreviated here, PBMCs. And we stimulate them with a combination of cytokines to grow dendritic cells. And dendritic cells are capable of presenting cancer-derived information such as epitopes and peptides on the surface. And they can show the antigen information to effector T cells. Therefore, we can give those dendritic cells back to patients so that in patient's body, dendritic cells can initiate the immune response against brain tumors. And a colleague of mine who is actually a specialist of dendritic cell biology had a nice analogy to describe dendritic cells. He said that the dendritic cells are the quarterback of the immune system. So when you talk about quarterback, in the place I live, many people think of this guy. So the reason why we think dendritic cells are quarterback, because of this. So dendritic cells, well, this is a cartoon I picked up from public domain information, but this is in general role. This is about the general role of dendritic cells in the immune system. So this is not specifically about cancers or brain cancers. However, there are lots of natural dendritic cells under the skin. So they are ready to pick up any infection information from the outside. Let's say in this case, maybe mucosa or skin. So there are viruses or antigen information that they can pick up. And as soon as they pick up the information, they migrate to the lymph nodes where they activate T lymphocytes. And the dendritic cells themselves, they do not have to go back to the site of infection. So it's usually T cells or B cells or NK cells, those effector cells. In the analogy of American football, they may be the running backs or wide receivers that really pick up the ball and go across the line. So in that interaction of antigen-presenting cells and T cells, there is a key molecule named major histocompatibility complex, MHC. So this is a general term for mouse and human, any kind of mammals. But when it comes to humans, the same molecule is called human leukocyte antigen, HLA. So that's the reason why some patients who contacted me about patients' eligibility, I always talk about HLA. So HLA is a molecule that is on the surface of antigen-presenting cells, such as dendritic cells, and also cancer cells. And there are many genes and proteins of HLA in everybody's body. In other words, every one of us have several different HLA molecules. HLA is one of those. And among HLA, there are dozens of different subtypes, A1, A2, A3, A24. The reason we picked up A2 is because A2 is most common in Caucasian populations. It's about 45%. So each HLA type molecule, they are different genetically, so their structure is slightly different. So they show the antigen information on their surface, but different HLA molecules show different peptides. So in other words, the peptides that can bind to HLA-A2 may not bind to HLA-A1. So therefore, we needed to design peptides that are able to bind to HLA-A2. And our peptide vaccine platform is composed of such peptides. Therefore, we need the patient to be positive for HLA-A2. So dendritic cells are the cells that are capable of doing this mission, I mean antigen presentation most efficiently. And dendritic cell was originally found by Dr. Ralph Steinman, who worked for a long time in Rockefeller Institution in New York City. And he received, in fact, this year's Nobel Prize for the discovery of dendritic cells, and he also demonstrated that the science can fruitfully harness the power of these cells to curb infection and other diseases such as cancers. And that was awarded October 3rd, so it's only 4 weeks ago. And however, 3 days before that, he passed away, in fact. So this information looks like a personal information, but I was very careful. This is from the public domain, so it's a public information from Rockefeller University website. But it's such a shame that we lost him. And the Nobel Prize committee did not know about his death until 3 days after, until the day they decided the award. So his Nobel Prize stands. But he is the one who actually discovered dendritic cells. Not only that, he has been a spiritual leader of dendritic cell-based therapy, cancer vaccine, and so forth. And he and I actually closely communicated, and particularly in the last several years, for for our common interest, and that is about the necessity of danger signals. So I told you about dendritic cells, and also told you in the beginning background slides that cancer cells, nevertheless, come from patients' own body. So it's not entirely unself. It's not entirely foreign substance. So cancer cells still a kind of self component of patients. So therefore, the initial trials of dendritic cell immunotherapy may not have succeeded because of the lack of sufficient what's called danger signals. So here comes the story of danger signals. In case of viruses, foreign substances, there's a clear molecular signature of such foreign nature. For example, some viruses have double-strand RNA. And we always talk about DNA and RNA, but a human body, our body doesn't make double-strand RNA. Our body makes single-strand RNA. So double-strand RNA, for example, is a unique molecular signature of viruses. And the immune system is so sophisticated and fascinating because the immune system has a specific receptor against double-strand RNA. And this receptor against, one of those receptors is called Toll-like receptor 3. There are many Toll-like receptors that recognize virus-specific or bacteria-specific molecular signatures. But the Toll-like receptor 3, for example, recognizes this double-strand RNA. So Dr. Steinman and independently myself have been developing strategies to provide such danger signals in cancer vaccine settings so that immune system can be fully activated. So during that process, we found the poly-IC-LC is a very attractive agent to that end. And this is provided by Dr. Andres Salazar in Oncovir. And this poly-IC-LC stimulates this Toll-like receptor 3 and other receptors that recognize double-strand RNA. And Dr. Steinman, in the last few years, have demonstrated that he tested a variety of different immunological adjuvants and says that Toll-like receptor 3 agonist poly-IC-LC is the most potent agonist. And independently, ever since 2007, we already found that in the brain tumor animal setting, and therefore we've been testing the combination of poly-IC-LC with vaccines. So what happens if you do a combination? So this is sort of like a highly scientific slide. I apologize if you are not familiar with those terms, but we looked at the lymphocytes. So if you talk about immunotherapy vaccines, your T cells have to go to the brain. But as I mentioned earlier, it's actually a challenge. Many people ask me during my seminar, hey, do T cells even go there? Even scientists and physicians ask me whether T cells can go to the brain. That's a good question. That's an important question because even vaccine itself, if you look at the T cells in the brain, we wanted to look at, in this case, cells in this grid. And cells in this grid indicates the vaccine-activated brain tumor-specific cells. And you don't get many, even though you give vaccine. And the poly-IC-LC itself doesn't do it. However, if you combine vaccine and the poly-IC-LC, you get lots of cells to the brain tumor side, and which is associated with the expression of brain-homing receptors on these T cells. And these are mouse brains. If you do vaccine itself, in this case, we used melanoma cell lines so that we can see those dark dots as the grown tumors in the brain. If you do this combination, you don't see those large tumors in the mouse brain. So this combination seems to be synergistically work. So based on these observations, we established our clinical trial platform, basically using multiple peptides of glioma-derived proteins, and sometimes dendritic cells, and sometimes peptides are combined with a chemical adjuvant that kind of replace the role of dendritic cells. And first of all, I'd like to talk about the phase 1 study, phase 1/2 study that was tested in patients with recurrent grade 3 or grade 4 gliomas. And this was published, so I'm going to go kind of summarized fashion. But the primary objective, we used dendritic cell type 1, very novel type of dendritic cells. There was no precedence of the use of same type of dendritic cells in humans before. Therefore, safety assessment had to be the primary objective. Nonetheless, the secondary objectives include immunological response and also preliminary anti-tumor clinical response as a secondary endpoints. And eligible patients had to be with recurrent WHO grade 4 GBM or grade 3 anaplastic glioma and HLA-A2 positive and minimum corticosteroid use. So that this can introduce a selection bias for patient populations, I have to say. But treatment, excuse me, yes, so treatment was with the ultrasound-guided intranodal injection of dendritic cells at 2 different dose levels, either 10 or 30 million dendritic cells per injection. And those dendritic cells were loaded with 4 different glioma-associated antigen peptides. One's coming from IL-13 receptor alpha 2, GP100, FA2, YKL40, and that was combined with what we call helper peptide. And the poly-IC-LC was given as intramuscular injections twice a week. So in summary, we treated 22 patients, and the regimen was very well tolerated. The common side effects were transient flu-like signs, and the poly-IC-LC induces interferon, and the interferon elicits flu-like signs such as low-grade fever, myalgia, fatigue transiently, which is not appreciated, but we see it as a biological sign of how danger signal is provided. And in terms of the dose escalation, dose-dependent effects, we did not see any dose-dependent effects on both safety and immunological activity. And we wanted to induce what we called type 1 immune responses in terms of the type of cytokine and chemokines. We actually did see very robust type 1 immune responses in all patients treated. And in terms of clinical efficacy, we saw 2 patients with the sustained complete response, including 1 patient with recurrent GBM. And 9 of 22 patients demonstrated progression-free survival of over 12 months. And we always want to learn what kind of markers or biological markers can dictate patient success. In terms of that, we found that the interleukin-12, which is a kind of cytokine, immunological cytokine, this level can actually correlate with the progression-free survival, as you can see here. So this is a graph showing interleukin-12 level from dendritic cells. The patients whose dendritic cells make high level of IL-12, interleukin-12, tend to do well clinically. And in terms of overall survival, this graph was plotted from the time patients entered our study. So considering that those patients already had recurrent high-grade glioma, so they had survived average about 12 months in case of GBM after the entry to our vaccine. So there is something interesting we learned. In the immunotherapy, well, this is particularly the case for immunotherapy, the tumor size can initially go up and then eventually go down. And we saw this pattern in several patients, and we find it interesting and important because we don't want to prematurely drop the patients off protocol. And interestingly, when I attend large-scale cancer immunotherapy meeting, in that doctors who treat melanoma patients, prostate cancer patients with immunotherapy, they all talk about the same thing. In fact, there have been publications talking about the necessity of new response criteria for cancer immunotherapy. So the typical traditional the traditional response criteria set for chemotherapy, they usually measure shrinkage of tumor immediately after the initiation of treatment. For example, WHO criteria for brain tumors, McDonald criteria that was modified from WHO criteria. We always talk about shrinkage. But as you can see here, as a representative case, in case of immunotherapy, because immunotherapy initially causes inflammatory response, on MRI scan, patient's tumor size can go up and down. And I could have updated this slide. I'm pleased to tell you that I saw this patient just maybe 3 weeks ago, September of this year, and the patient tumor is still sort of like a gradual shrinkage or a stable stage. So this patient with recurrent GBM, with no combination with any other treatment modality, he had been progression-free for over 2 years by now. And another kind of lesson is when you see new enhancement like this. So this is another patient in that we vaccinated, and eventually we saw new enhancement. So we thought this was a failure. We took the patient down to the OR, a neurosurgeon rejected it, an independent neuropathologist looked at the tissue and says that this is not a recurrence. There's no evidence of tumor cell mitosis. Instead, he saw a lot of immune cell infiltration in the lesion that was rejected. So this is a typical example of pseudo-progression. And so telling us an implication, lots of implication about a necessity of new criteria or a new method of assessment for immunotherapy therapeutic response. Also, I'd like to switch my gear a little bit towards low-grade glioma vaccine. For many reasons, I think vaccine therapy can be most appreciated in patients like low-grade glioma. And so we developed this low-grade glioma vaccine pilot clinical trial. This is supported by, I need to mention also the previous trial, high-grade trial and low-grade trial. Both are supported by NIH as well as Mosela Foundation. And this low-grade glioma trial, we developed with the doctors in Wake Forest University, particularly Dr. Ed Shaw, who is an internationally well-known expert of low-grade glioma treatment. And so there are lots of reasons we think this is important. So low-grade glioma patients, the tumors, most cases sitting for a long time or growing very slowly. So we have plenty of time. Vaccination takes time. In case of chemotherapy, as soon as patients get the drug, it starts working. But vaccinations, you need a long time. You need to repeat vaccinations, and you need to see this immune response really kicking in and activate the system. But in case of, often in high-grade gliomas, because of the rapid growth, we do not have sufficient time. On the other hand, low-grade glioma cases, most cases, we have sufficient time. And the minimum toxicity associated with vaccine allows for maintenance of high quality of life in those patients. And also, in those patients, chemotherapy is not the standard of care, and the optimal timing and dose for radiation has not been determined. So often cases, patients are just left off without any active treatment. We may be able to mount protective immune responses in low-grade glioma patients. We initially had the criteria, particularly selecting high-risk low-grade glioma patients. This was mandated by NIH review group and so forth. However, after treating 10 patients safely, now we have been able to lose up the eligibility criteria a little bit so that we can take oligodendroglioma glioma patients and also oligoastrocytoma patients without particular risk factors. I wanted to show this slide one more time because I wanted to show the list of peptides, antigens. So we use synthetic peptides, not patients' own tumor cells. And those antigens, the vaccine antigens were selected from proteins that are most commonly overexpressed in high-grade gliomas. There is some expression of these in low-grade gliomas, but in general, these proteins are much more expressed at much higher levels in high-grade glioma patients. Here is a notion. So what we are doing is not only therapeutic vaccine, but the preventive prophylactic vaccine. So the clinical issue in most cases is that low-grade glioma patients, particularly in adults, more than 50% of cases, in the past at least, what have been seen is that recurrence with high grade. So we'd like to mount an immune response against high-grade glioma cells when patients are still with low-grade. So the notion is more like a prophylactic vaccines. And the last portion of my clinical trial, I'd like to mention a little bit about the pediatric version of our clinical trial. So our study, basic laboratory studies have shown that the pediatric gliomas, despite we all know that pediatric gliomas have distinct biological features compared to adult gliomas, nonetheless, those pediatric gliomas, most cases express similar antigens as adult gliomas. So we have been able to use the same combination of antigen combinations and platforms for pediatric patient cohort. So we've initiated pediatric vaccines, particularly brainstem, diffuse brainstem glioma patients. So here's one of the cases. We saw similar cases, by now several cases. 10-year-old girl with the typical DIPG here, diffuse infiltrating pontine glioma. And this is at the time of diagnosis, May 2010. And after radiation therapy as a standard of care, we saw this. This is before vaccination. And we had about 3 months from the completion of radiation and initiation of vaccine. However, 3 months after the initiation of vaccine, that was about Christmas time of last year, we saw this. If you compare with the previous slide, so now, unfortunately, the tumor looks larger, and the patient condition was worse. And it was before Christmas. Unfortunately, we gave up, and the parents decided to send her to hospice. And however, after the holiday weeks, in January, they called us up and said that the patient is doing much better. And what was done in hospice stay was only steroid treatment. And my colleague, Ian Pollack, who has been seeing pediatric gliomas for 20 years, says that he has never seen such a dramatic shrinkage of pontine glioma. And so looking back, it was an inflammatory response induced by the vaccine that was making the tumor look much larger. In other words, if that was there had been a true progression, we wouldn't have been able to shrink only by steroid that much. So this patient is still around and beating the odds. So she's been surviving a year and a half after diagnosis, and we monitor immune responses. And it was interesting, when we started steroid, high dose steroid, the immune response actually shrinks down. But there was a trend showing that the immune response is bouncing back after she was put back on vaccine trial. So we've been going through lots of winding road, and this has been no man's land, and actively vaccinating patients. And we've been learning a lot. And so we have some conclusions, however, particularly the second point. Novel peptide-based vaccine demonstrated tumor pseudo-progression followed by disease stabilization or shrinkage as a part of the treatment effects in multiple cases, including pediatric cases with brainstem glioma. So I hope I'm not taking too much time, but let me spend a few minutes about what I think is a dream concept, which is available in a matter of few months. So let's talk about current issues in the current form of glioma vaccine. So I talked about some successful cases, but I also have to say that there are some cases that didn't respond. So why we see some cases responding, why other cases not responding, that's very important to address that. Maybe the reason is that because we use universal vaccine, so we treat all patients with the same vaccine. So we select 4 antigens that are most commonly expressed in patients' glioma, many cases of glioma tissues, but you never know whether each patient's tumor really express any or all of targeted antigens. We evaluate this as much as we can by taking tumor samples and so forth, but the use of universal vaccines always has a risk and potential downside of not really meeting individual tumor characteristics. On the other hand, there is actually personalized vaccine approaches. Many investigators use patients' own tumor lysate. So those vaccines can be considered personalized vaccine because they use patient's own materials. If the patient did not get sufficient tumor tissue amount, they may not be eligible. But those can be considered personalized vaccines. However, there's a risk, theoretical risk about it. So brain tumor cells still express a lot of proteins that are also made by normal brain cells. Now, so far in the field, we haven't seen many cases of autoimmunity against the brain itself. But as we advance, we are advancing the field, and we have started using more and more potent immunologic strategies, such as novel adjuvant, poly-ICLC, and so forth. As we advance our technology, we may actually start seeing autoimmunity if you use bulk tumor materials, because a whole tumor can still express normal brain cell components. So the ideal vaccine should reflect each patient's own gene expression and mutation status, and yet not including antigens from normal brain cell components. So how can we achieve that? So I started collaborating with the very ambitious and great group in Germany. They originally stemmed from University of Tübingen, and they formed a biotech company named Immatics. And they developed a consortium mostly with European academic institutions. And I am going to be the only participating investigator in the United States. So this consortium has proposed an approach called Glioma Actively Personalized Vaccine Consortium, GAP-VAC. So what they do, so let's talk about personalized medicine. So personalized medicine is an intense focus in the current oncology drug development. In the past, two levels of personalization are discussed. Number one, the biomarker-guided approach. Say, HER2-neu positive, patients for HER2-neu positive tumors, there is a therapy. And EGFR positive glioma vaccines. In my case, patients have to be HLA-A2. And the downside of this is that if you are not positive, if you are negative, you are left off. You are forgotten, basically. That's not good. I know it's not good. So on the other hand, as I mentioned earlier, passive personalization approach. For example, the use of whole glioma cell lysate. But as I mentioned a minute ago, there's a risk of inducing autoimmunity because of the presence of normal brain cell components in the whole tumor lysate. So let's seek more actively personalized approach. So we're going to have to use biomarker-guided manufacturing of a vaccine specific for each individual patient, taking advantage of sophisticated analysis of genomic, proteomic, and peptidomic molecular biomarkers. This was originally proposed by my German colleagues, German Consortium Association for Immunotherapy. So we have to give them a lot of credit for this concept. What they do is actually to combine for each patient two different classes of peptides for vaccination. One group is a group of peptides that are overexpressed in tumor tissue of each patient. So this is a quantitative difference, so not qualitative difference. The same protein, if the tumor cells make 100 times more of the same protein compared to the normal brain, it may be considered specific. So let's take some of those overexpressed protein and the peptides from those proteins for vaccination. Still, that analysis has to be made in individual basis. And also, tumor, we all know that cancer cells contain a lot of gene mutations. One can say it can be up to hundreds of mutations that one cancer cells can contain compared to the normal cell from the same individual. So gene sequencing technology has been advancing quite a lot. And now we are talking about each patient's personalized gene sequence from each patient's tumor tissue. And even up to 3 years ago, I thought it was a dream. It's easier to talk. Everyone can conceptualize such a thing. It's much harder to implement. But just like any other cutting-edge technology, if you wait 3 years, cost always comes down, feasibility goes up. Now we can talk about feasibly about implementing such a strategy. So what we can do is to sequence each patient's tumor tissue and find specific mutations, and the mutation can contain lots of immunogenic peptides because mutation is specific in tumor cells. So the healthy cells do not have the same mutation, therefore immune system can recognize this mutation spot as non-self. So they can have much higher, they can demonstrate much higher immune response against mutated spot. So we have already have a concept of clinical trial, and we think that within 5 months, we can do gene sequence and also peptide analysis in order to identify patient-specific gene mutation, specific peptides, and make peptides for vaccines within 5 months. So 5 months can be shortened in the future, near future, when technology further advances. During the waiting period, let's say after diagnosis for newly diagnosed patients, HLA-A2 positive patients can still get ready-made universal vaccine, just like we've been doing already. So in terms of schema, I just mentioned newly diagnosed scenario. In the scenario for newly diagnosed GBM patients, they can be recruited, and tumor material will be analyzed for tumor mutation, tumor-associated overexpressed or mutated peptides. And median progression-free survival in earlier trials of radiochemotherapy, in other words, current standard, has been determined to be 6.9 months, in that about 70% of patients are still stable at 5 months. So if we implement our current technology, we think we can achieve the vaccination in 70% of patients before they recur. And we plan to treat 30 patients. So this is the kind of schema of our treatment plan. So here's the last time I show HLA-A2 positive versus negative sort of strata. And patients who are positive for HLA-A2 during and after radiation and temozolomide standard of care therapy, they can start receiving vaccinations with ready-made off-the-shelf universal vaccine that was already made for HLA-A2 patients. And for negative patients, they can still take the standard of care medication. But within 5 months after the diagnosis, we'd like to start vaccinating them with the truly unique, truly personalized, actively personalized peptide vaccines. So 2 last slides. So personalized medicine can be achieved by stratification of patients according to biomarker method predicting therapeutic benefit. Ideally, this would mean that the grouping of patients to different kinds of therapies with all of the patients profiting from the respective treatment. So far, to date, that is not the case. Again, I feel responsibility for selecting HLA-A2 patients for my current vaccine, but that's not ideal. In reality, treatment is often restricted to the biomarker-positive patient population. Patients who are not eligible for the available treatments are neglected. Additionally, few disease-specific features are addressed by current biomarker-driven therapies. And in malignancy, inclusion of tumor-specific features as a target for therapy is particularly interesting since tumors evolve differently under the distinct evolutionary pressure in each patient. In other words, cancer-specific mutations usually are not found. Those are not present. Those are not found in normal cells. So we can identify truly unique molecular signatures in the tumor cells in individual basis. And so GAP-BAC would be the first multi-center trial globally where immunotherapeutics will be actively and specifically designed for individual needs of patients, and therefore will take personalization to the next level and amplify clinical benefit of patients. So I'm stopping here. Well, thank you for staying with me, and I'm happy to take any questions. Thank you for your attention.
You're talking about taking tumor tissue. A lot of patients have their surgeons at local hospitals. They like their surgeons. They want to deal with their surgeons. But I've heard horror stories about they get the surgery, that tumor tissue has to be taken immediately, frozen, and sent to wherever it has to go to make up this vaccine. And you hear that the hospital won't release the tumor tissue, the patient loses it.
Right.
What do you do? How do you obtain tumor tissue and make sure you get it?
Right. So, currently ongoing studies utilizing patients' own tumor tissue, they need tumor lysate. So they need, in other words, they need tumor cells and tumor proteins. So in that sense, it's very different from the approach I'm proposing. In the approach I'm proposing, we're going to need genetic information of the tumor cells. So usually gene sequencing requires much less amount of cell materials compared to the amount that would be necessary for lysate-based vaccines, because the beauty of genetics is that you can always amplify, basically. And the lysate, tumor lysate, there's no way to amplify the amount of lysate that you obtain from surgery. Well, there is. I mean, you can grow those cells in culture, but once you put them in the culture, their phenotype and their protein expression profile can be completely different. On the other hand, genetic approach, we do not need much amount of genes for completing the gene sequencing. So we are carefully optimistic about the feasibility of our approach. And still, though, the infrastructure level, in order to obtain the tumor tissue material from the hospital in that patient was operated, that's still, I agree with it, it's a considerable issue that needs to be improved.
So what do you do about it?
Yeah, as a participating center, well, we probably have to have patients operated in the participating centers, in any of those clearly participating centers, for example, University of Pittsburgh, to participate in this vaccine, because that is the best way to ensure the quality control and quality assurance of the tumor material for the sequencing. So initially, we're going to have to do that. But again, I'm feasibly optimistic. Those technologies and gene technologies can evolve, and the amount of DNA material for gene sequencing has been decreasing and increasing. In other words, we can deal with much less amount of gene material to get the same amount of information compared to 10 years ago. So therefore, I believe that the feasibility issue will probably step by step will be resolved or be improved.
Don't they have a company somewhere in New Jersey, I don't recall the name of it, that contacts the hospital, works with the legal system of the hospital, and guarantees the tumor? I mean, you have to set this up in advance. Isn't there a company that does that?
I have heard about it, yes. Yes.
But you don't know the name?
I'm not in a position to comment about their feasibility or as to whether we can work with them immediately for this project. I cannot comment yet because I'm not familiar enough with the system.
Then I had just another question. Has to do with the different types of dendritic cell systems.
Right.
There are some companies that are working on, instead of going after the tumor, going after the cancer stem cell, because they claim that going after the tumor, you may be able to kill the tumor, but the actual cancer stem cell keeps generating new cancer cells to create new tumors. So instead of going after the tumor, go after the root. It's sort of like cutting off the top of a weed, and then it keeps growing back again.
I can comment on with two lines. For two aspects. One is that, in fact, most of the antigens we currently target, are also expressed in cancer stem cells, such as glioma stem cells. So if you really target most aggressive population of glioma cells, you are likely to be also targeting glioma stem cells because there's no clear line of distinction between the really aggressive glioma cell populations and glioma stem cells. I mean, initially several years ago, glioma stem cells created a big sort of, that was a buzzword. And then we were certainly excited and we pay a lot of attention to it. But as we learn more about it, we also learned that there may not be as a clear distinction between relatively aggressive glioma cells versus stem cells, what we call stem cells, and markers that was originally proposed to distinguish between stem cells and versus non-stem cells. There has been lots of controversy about it now. And my general consideration is that if you really carefully look at the genes that are responsible for glioma cell survival, glioma cell growth, you are also likely to be targeting glioma stem cells. I forgot to mention that in the process of gene screening, there's the algorithm for selecting most important genes. Not only by the quantity, not only by the mutation. We also select ones, when you come up with 50 genes, for example, of mutations, or 50 genes that are relatively overexpressed in gliomas. Among them, biologically, we select ones that are likely to be responsible for glioma cell survival, or glioma cells growth. Because if you target those genes, you are likely to be targeting cells that are most aggressive, cells that are most responsible for the survival of the tumor tissue. So we naturally incorporate, I appreciate your keenness on glioma stem cell biology, but in slightly different ways, we seem to be addressing the same issue in our algorithm.
How feasible is it in the case of a diffuse tumor that you can get good enough sample to develop a personalized vaccine?
Yes. There are, so diffuse tumors, the surgical, your question can be also addressed in cases that surgical resections are not really feasible. Right, so those are really still challenges. However, I didn't mention, but through these efforts, by analyzing, let's say, 30 individual cases for their gene profiles and expression profile and relatively commonly mutated peptides, we also aim to come up with a warehouse, what we call warehouse of peptides. So in order to make my message clear, I sort of simplified my talk in a way of total personalization. But at the same time, we are interested in establishing more sort of better selection of shared antigens that can be commonly used for patients who may not have tumor tissue.
Thank you.
Sure. Yes.
This will be the last question because we're running out of time. If the HLA-A2 positive vaccine works, is there any reason you couldn't make a similar vaccine against, say, like HLA-A1 positive or A3 positive?
Yes. For this last approach, we don't limit the patients.
No,
to HLA.
not that one, for the previous
but
one.
that sort of represents the effort, our effort, with using the similar technology, carefully analyzing the immunological epitopes. Yes, we can extend our library of peptides to, and from currently we only have almost HLA-A1 or A2 only, but we can expand our library of peptides for other HLA haplotypes, certainly.
Okay, thank you.
Yeah. Thank you.